Welcome to the deep dive. Our source material today takes us into, well, the cutting edge of conservation genetics, a fascinating paper from Molecular Ecology Resources.
That's right. It's titled Estimation of Species Abundance Based on the Number of Segregating Sites Using Environmental DNA, eDNA, and it's led by Yuan AI and team.
And this research tackles really one of the biggest bottlenecks in biomonitoring, doesn't it?
Absolutely. Environmental DNA, eDNA, it's been revolutionary for detecting species, answering that basic question, is it there? Think endangered species, invasive species monitoring.
Yeah, huge for that.
But the real holy grail, the thing everyone's been working towards, is moving beyond just detection to accurate quantification.
Right, estimating exactly how many individuals are actually in that population.
Exactly. That's been the challenge.
So our mission today is to understand this fundamental shift, how these researchers developed a method that might dramatically improve our ability to count species using just that genetic trail left behind in the environment.
It's all about finding a reliable, non-invasive way to do a census. And the traditional approach, just measuring the total amount of DNA, well, it turns out it's fundamentally flawed once you step outside the lab.
Okay, so let's unpack that flaw. If this DNA is just shed into the water, why has simply measuring the concentration, the sheer amount of DNA, proven so unreliable for counting, say, a population of fish?
The main issue is variability, and it comes from, well, multiple places, biology and the environment. The authors talk about what they call differential individual DNA contribution.
Meaning, not every fish is shedding DNA at the same rate. Is that the idea?
Precisely. Think about it. You've got factors like the life stage of the fish, its physical size, how active it is, even if it's just eaten.
Oh, okay.
All of these can significantly change how much DNA an organism releases per hour, per day.
Yeah.
You might have one large, super active fish shedding, I don't know, 10 times the eDNA of a smaller resting one.
If you're just counting total DNA.
Right. That one hypershedder could completely skew your results. It might make you think there are 10 times more fish than there actually are. It leads to potential overestimation.
And that's just the biological side. If those variables weren't enough, the environment itself is constantly working against you.
Absolutely. The DNA itself doesn't just hang around forever. Decay rates are very sensitive to things like temperature and UV light.
Okay.
Plus, you have physical transport, water flow, currents. They move the DNA around, so the concentration you measure in one spot might be more about where the water carried it than how many fish are upstream.
So all these factors, biological and abiotic, just muddy the water, so to speak.
Exactly. And that's why previous studies using just concentration have been so inconsistent. Some reported strong correlations in controlled settings, sure, but then others found non-significant results out in the wild. We desperately needed a metric that's more resilient to all that noise.
Okay, so here's where the innovation really kicks in. The researchers pivot. They move away from quantity, the amount, and look at genetic diversity instead. They propose this new metric, the number of segregating sites. Explain that for us. How is this different from just counting DNA molecules?
Okay, so a segregating site is essentially a polymorphic site. It's a specific point in the DNA sequence where individuals within that population have different base pairs. Genetic variations.
Right.
So instead of measuring the sheer bulk of DNA, we're measuring the number of unique genetic variations we detect. Think of it less like counting all the coins in a jar, which is like concentration, and more like counting how many different types of coins or different currencies are in that jar. That's kind of like segregating sites.
That's a great analogy. So back to the hypershedding fish. It might drop 1 million DNA fragments, inflating the coin count, the concentration.
Yeah.
But those million fragments still only represent one individual's unique set of variations.
Exactly right. This whole approach uses the inherent redundancy of genetic markers. Whether an individual sheds 10 copies of their mitochondrial DNA or 1,000, the unique genetic variations that define that individual's contribution, they effectively get counted only once when you tally up the distinct segregating sites.
So the metric itself is just inherently less sensitive to those chaotic, uneven contributions. It sort of filters out the shedding noise.
Precisely. It's designed to be more stable against those biological fluctuations.
This really does feel like a fundamental shift in how we think about eDNA quantification, which of course raises the critical question about testing it. How did they rigorously check if this elegant theory holds up under real-world messy conditions?
Yeah, that's key. They used a remarkably thorough three-tiered approach, moving meticulously from perfect theory towards a controlled but complex natural context.
Okay, walk us through it. Starting with Tier 1. in silico. Simulated data.
Right. First, they needed to establish the basic genetic relationship in a perfect world, you know? They simulated populations, about 1,000 individuals, with fairly long DNA sequences, around 17,000 base pairs.
Why that length?
It approximates the size and information density of the mitochondrial genome. That's a common target for eDNA because there are lots of copies per cell. They tested this simulation at different mutation rates and confirmed, yes, in this ideal digital environment, the number of segregating sites scales very nicely with the number of individuals. R-squared values were consistently above 0.9. Strong correlation.
Okay, proof of concept digitally.
Yeah.
Then tier 2. In vitro, using real tissue samples.
Yeah.
How do they bridge that gap?
So they moved to actual biological material. They used tissue DNA from 2 brackish gobies species, Acanthogobius hosta and Tridentiger bifasciatus. And they used a crucial bit of tech here, target enrichment.
Explain that.
They designed universal RNA baits based on conserved mitochondrial DNA regions found across vertebrates. These baits let them efficiently fish out just the mitochondrial DNA sequences they were interested in from the total DNA mix.
Got it. Like a molecular magnet?
Kind of, yeah. Then they simulated an eDNA mixture by randomly sampling sequences from these known individuals. And again, they confirmed a strong positive correlation between the number of segregating sites, SS, and the known number of individuals they'd sampled from. For one species, A. hosta, the R-squared was incredibly strong, like 0.98.
Wow. Okay, digital world checks out, lab bench checks out.
Yeah.
Now for the acid test, tier 3. In situ, the mesocosm experiment. This is where they deliberately introduce the noise, right?
This was absolutely the make-or-break phase. They set up 20 controlled aquarium environments, mesocosms, using one of the goby species, A. hosta. They systematically varied the number of fish in each tank, 7, 14, 21, 28, 35 individuals. But critically, they also introduced those confounding factors we talked about earlier. They deliberately manipulated the average body size of the fish in different tanks, and they compared tanks where fish were actively fed versus tanks where they weren't feeding.
Trying to mimic that real-world variability.
Exactly. Then they collected the eDNA from the water, performed the target enrichment and sequencing, and compared the performance of their new segregating sites metric against the old standard. eDNA concentration measured by qPCR.
So the moment of truth. Once they threw in those real-world variables, size differences, feeding activity, what did the results show? Did the segregating site metric actually hold up better than concentration?
Unequivocally, yes. It was a really clear result. Now, both metrics did show a statistically significant positive correlation with abundance. So you can predict population size to some degree with the but the difference in precision was striking.
Give us the numbers. How much better was it?
Okay. So the actual number of fish in the tank, the species abundance, predicted the number of segregating sites with an R-squared value of 0.7.
70% of the variance explained. Okay.
Exactly. Now compare that to the traditional concentration method. Species abundance only accounted for 46% of the variation in eDNA concentration, an R-squared of just 0.46.
Wow. 0.46 versus 0.70? Yeah. That's a huge jump in predictive power.
It really is. In a controlled but deliberately noisy environment, the SS metric explains significantly more of the variance. It's just a much more accurate and robust way to estimate the numbers.
That difference alone is massive for anyone trying to do monitoring. But what about those specific biological variables they added? Did the fish size or feeding behavior still mess with the segregating site count?
And this, I think, is the most crucial finding, especially for practical long-term monitoring. Those confounding variables, the mean weight, so body size, and the feeding behavior, they showed no significant influence on the number of segregating sites. The p-value from the F-test was 0.38.
No significant influence. That's the resilience signal right there, isn't it?
That's exactly it. For a field researcher, knowing your estimate isn't going to be thrown off just because some fish are bigger or just ate. That's invaluable.
And how did concentration fare against those factors?
Well, in contrast, those same factors, size and feeding, showed a nearly significant effect on the traditional eDNA concentration. The p-value was 0.07.
Close to that 0.05 significance threshold.
Right. It wasn't strictly significant by convention, but it's dangerously close. And it perfectly illustrates how sensitive that concentration method is to just the day-to-day biological fluctuations. The SS approach just effectively filtered out that noise.
And I remember reading about the cross-validation tests, too. They further supported this advantage.
They did, yes. When they tested how well the models could predict abundance using subsets of the data, the SS approach consistently showed lower absolute bias, meaning the estimates were closer to the true value on average, and it showed reduced standard deviation.
So more precise estimates as well, less variability.
Exactly. The estimate was not only more accurate, but also much more consistent from sample to sample. And that advantage was particularly clear when they used smaller training data sets, which is often the reality when you start a new field study.
Okay, let's connect this to the bigger picture then. This SS method seems superior in accuracy and resilience. But it sounds like the analysis is, well, potentially more complex than just running a quick qPCR for concentration. Does this method, needing high-throughput sequencing and targeted enrichment, sacrifice cost-effectiveness or ease of use?
That's a very important practical question. And yes, while using segregating sites directly might actually simplify the downstream bioinformatics compared to trying to assemble full haplotypes or run complex mixture models.
Okay.
It certainly requires more upfront sequencing effort than a simple qPCR rate. There's no denying that. However, the researchers make a strong case that using target enrichment, especially with baits designed for conserved regions like mitochondrial DNA.
The ones that work across many vertebrates.
Right. That makes the approach potentially multi-species applicable using the same set of baits. That could actually save significant time and money compared to designing and validating species-specific qPCR primers for every single target.
Ah, I see. So the initial sequencing cost might be higher, but the accuracy gain and potential for broader application could justify it, especially for really critical conservation projects where getting the count wrong has serious consequences.
That's exactly the calculation, I think. A high-precision census, even if it costs a bit more upfront, provides a far better foundation for management decisions than a cheap but potentially very inaccurate one.
Makes sense. Okay, let's turn to the necessary caveats. What do researchers need to keep front of mind if they want to take this method out of the mesocosm and into, say, a real river or a lake?
Well, the primary caution is just that. The crucial next step, large-scale, real-world field testing, hasn't been completed and published yet. These fantastic results are still based on controlled, albeit complex, environments. Real-world hydrodynamics, water mixing, sediment interactions. It's orders of magnitude more complex.
So, results need cautious interpretation in the field for now. What else?
Secondly, there's a tricky technical challenge in defining the threshold for actually counting a true segregating site. You're digging through potentially millions of sequence reads, trying to distinguish real, often rare genetic variations from inevitable sequencing errors.
Right. Errors happen in the sequencing process itself.
Exactly. And there aren't really global standards for this yet. The researchers here used what's called an ad hoc filter. They only counted variations that appeared in more than 7 reads and had a frequency above 3% within their sample.
But setting that filter is a balancing act, isn't it?
It really is. Set the threshold too high, you risk missing true, rare genetic diversity, which would underestimate the population count, especially in diverse populations. Set it too low, you risk inflating your count with sequencing noise, making you think there's more variation and thus more individuals than there really are.
So how do we get better at distinguishing the signal from the noise there?
Well, the future likely involves incorporating more advanced sequencing techniques, things like duplex sequencing, or maybe more practically in the near term, using unique molecular identifiers, UMIs.
UMIs.
Yeah, these are like unique barcodes attached to each individual DNA molecule before any amplification steps like PCR. They let you track each original molecule through the whole process. So if you see the same variation tagged with multiple different UMIs, you can be much more confident it's a true biological polymorphism, not just an error that got amplified many times.
Ah, okay. A way to digitally fingerprint the original molecules.
Pretty much. It helps ensure you're counting unique information, not just copies of noise.
And there was one final caveat mentioned about needing a baseline survey.
Yes, that's important. This method isn't plug and play with a universal calibration curve. It requires a pilot study for your specific target population to establish that baseline relationship between the number of segregating sites and abundance in that context.
Because the background level of genetic diversity varies between populations.
Exactly. And if you get that initial characterization wrong, if you misspecify the genetic variation in your pilot study, it will bias all your future estimations based on that curve.
How do they suggest managing that?
Well, Cleverly, they suggest using the same kind of in silico modeling they did in Pier 1. You can use known parameters, like estimated mutation rates for your species group, to simulate and figure out the optimal number of individuals you need to sample for an effective pilot study. It helps manage resources, ensuring you capture enough diversity for a good baseline without massive oversampling.
Right, planning the pilot study intelligently. So overall, this study really does mark a significant advance, moving eDNA quantification towards, well, precision and accuracy by focusing on genetic information content rather than just bulk DNA.
Absolutely. It proposes the number of segregating sites as a far more resilient proxy than simple concentration. Moving forward, the community will be keenly watching for those next steps. Extended testing across more species, different environments, and critically, those real-world field validations, and refining methods to confidently identify those true low-frequency alleles in messy eDNA samples.
So boiling it down, what does this all mean for you, the listener, whether you're working in biomonitoring or just interested in how we track life on Earth?
It means the future of counting wildlife, particularly elusive aquatic species, could rely less on sort of how many total DNA molecules we happen to scoop up, and much more on how much unique genetic information we can reliably extract from the molecules we do find. It holds the promise of delivering reliable, high-precision population counts non-invasively, maybe for the first time in many contexts.
Okay, time for our final provocative thought for you to mull over. The study suggested differential DNA shedding is quite erratic over short experiments. So could doing longer field studies actually worsen the predictive power of the old concentration-based methods, as those confounding factors like uneven shedding, decay, transport accumulate over more time? Could that make the performance gap between concentration and segregating sites even wider in the real world than the 0.46 versus 0.70 they found here?
That's a really interesting point. It raises this compelling possibility that the benefit of using a genetically resilient metric like segregating sites might actually increase the longer and more dynamic the monitoring period is. The noise just gets worse for concentration over time, while SS stays relatively stable.
Something to think about. We hope this deep dive into genetic variability and abundance estimation gives you a clearer view of both the challenges and the really exciting cutting-edge solutions emerging in non-invasive conservation science.